Hi everyone, it's Belinda, and this is our Chapter 1 Biology, the Study of Life lecture. This section has 4 sections, and this chapter is on Exam 1. So the first thing to look at here is that these are our objectives. And if you want to take a moment, you can even pause this video and you are going to rank yourself with your knowledge on these objectives. So start with define biology, give yourself a score of 1, meaning you have no idea what that is, to 5, you think you are an expert, and do that for our other objectives. Then come back after this lecture section and rescore so that you can see where your knowledge has improved and maybe the part that you need to go back and review. So let's start with first the characteristics of life. So biology is the study of living things or the study of life, and there is a great diversity amongst living things. So you can do a little quick exercise here looking at this picture. Make a very quick list of all of the living things that you can imagine. And maybe you've made a list that includes bacteria or protists or fungus, different plants, different animals, and all of those things would be studied under biology. There's also a great diversity in living things because all living things share some basic characteristics. Living organisms, or that individual, are made of the same kinds of chemical elements that non-living things are made of. So we're all made of atoms. And these living things have to obey the same physical and chemical laws that govern everything else in the universe. So something like gravity still applies to a living thing. Life is very organized, and there are levels of organization going from atoms, which will be our very smallest unit of matter, all the way up to our biosphere, which is anywhere that a living thing can be on our planet. A cell is the most basic unit of living things. It's what has structure and function for all living organisms. And a living thing can be unicellular or just have one single cell that makes up the whole living thing, or it can be multicellular, having many cells differentiated to different jobs that all work together. And each level, moving from atoms to molecules all the way up to that biosphere, is more complex than the level that happened before it. And as we increase that complexity, there's new properties. That's why it's a new category. So let's take a look at those categories. First, we have atoms. Atoms are the smallest unit of an element. They're made of electrons, protons, and neutrons. And an example might be something like oxygen. A molecule is two or more atoms that are combined together. So we may have carbon and oxygen, or we may have carbon and hydrogens, and we could make something like methane. We can make really large molecules. We may even call them biomolecules, like a protein. Then we have a cell. Again, our cell is the basic structure and function unit of all living things. And again, living things can be single-celled or multi-celled. If I have a group of cells that do the same sort of job, then that would be called a tissue. So for example, I could have a lot of nerve cells, and then that becomes nervous tissue. If I then have a bunch of different tissues that work together, then I can have an organ. For example, I could have nervous tissue and epithelial or skin type tissue, and I could have fat or adipose tissue all working together for a job, and then that organ is called the brain. Organs that work together are called organ systems. So I may have something like the brain, spinal cord, and nerves. And then that's the nervous system. And then if I have a bunch of different organ systems that are all working together to do a job, then that is an organism. So you can have a nervous system and a cardiovascular system and a muscular system and a skeletal system. That makes you the one singular organism. If I have a bunch of the same kind of organism all in a place, then I could call that a species. If I then look at that species all in the same place at the same time, then that is a population. If I am looking at that species with other species in a group, then I am looking at a community. If I'm looking at multiple kinds of communities here with our different animals and plants, but I'm also looking at the not living parts of the environment, I call it an ecosystem. Any big part of our planet that has a similar ecosystem, we may even call it a biome, something like a desert or a tundra. And then last but not least is our biosphere. That's all the air, water, and land where living things can be found. So now we have a chance to check your knowledge based on what you just were presented. So again, you may pause this video, but a group of similar individuals that can interbreed would be considered what? And that should be B, species. Question 2. All of the populations of different species living in one area would be categorized as, and that should be C, community. Now we have a chance to take a look at all of these photos, and you may want to pause the video. Can you categorize these things as alive or not alive? And why did you put it into that group? If we take a look, we can see that this mushroom and this snail, they're alive. Compared to a rock, not alive. Compared to a cow, is alive. Compared to a computer, not alive. Well, why are computer and rock not alive and cow and mushroom alive? Well, maybe you thought of things like they grow, or they breathe, or they adapt over time, or they have cells. And all of those would be really good starting points for our next part of our presentation. So we want to take a look at the 4 basic characteristics common to all living things. The first is that life requires material and energy. The second, living organisms respond and maintain an internal environment. The third, living organisms reproduce and develop. And the fourth, living organisms have adaptations. And we're going to go ahead and take a look at each of these a little more in depth. So number 1, of course, was life requires materials and energy. Energy in a science definition is the ability or the capacity to do work. And energy is required to maintain that organization, right? From an atom to a molecule to a cell and so on. That requires energy. Energy is also required to conduct life-sustaining processes like chemical reactions, such as breaking down our food and then giving us those atoms to use to build our bodies. Metabolism is the vocabulary word we use for all chemical reactions that are occurring within a cell. If we then look at this energy sort of top down from an ecosystem is characterized by chemicals and energy flowing through it. because chemicals are not used up when organisms die, but they're moved from one population to another in a food chain, right? Something will eat something else, and then they're gaining those chemicals. For us humans, right, you'll eat a turkey sandwich or you'll eat a salad, and you're gaining those chemicals. And then chemicals will move from producers or the things that make their own food. And then that will go to the consumers. Those are things that eat the producers. And then to decomposers. Decomposers break down dead producers and dead consumers. As a result of death and decomposition, those chemicals are put back into the soil for our plants to use. Energy also moves in the ecosystem. So we have sun that is going to flow to plants that will use that solar energy to make sugar in photosynthesis. And then that energy can move to consumers when they eat the producers. And then, of course, that energy will go to decomposers that are breaking down dead consumers or producers. What we also notice is that a lot of that energy is lost as heat to the environment. So there has to be constant solar energy input in order for our ecosystem to have enough energy. The second part is that living organisms maintain homeostasis and respond to their environment. Homeostasis is that internal balance within certain boundaries, and it's very important for a living organism to keep a steady state or a biological balance. And a feedback system helps us to monitor. So if it goes beyond that boundary, we bring it back down. That's that feedback system, and it's able to help us make adjustments to maintain homeostasis. Living organisms interact with their environment and respond to those changes. That's responsiveness, and it is part of this maintaining homeostasis and responding to the environment because we have to be able to react to changes. And the ability to respond to our environment produces a movement. So there's a nice little quick check here. You may want to pause the video. Think about what happens when you reach for a pot on the stove and it's hot, right? What happens? Can you think of other kinds of responsiveness? Maybe you thought of things like anytime you smell food, you start to salivate and your stomach starts to growl. Or maybe you thought about when you kick a ball with one foot, that your weight shifts to the other leg so you don't fall down, right? All of those would be responding to what's happening in our environment. Number 3 is that living organisms reproduce and develop. All living things must reproduce to keep their population. The manner of reproduction will vary. There is asexual reproduction where we do not require two individuals to share their genetic material. And then there is sexual reproduction where we must share that genetic material. When organisms reproduce, they pass on copies of their genes to the next generation, and the genes will determine the characteristics of the living thing. Genes are made of DNA, and the big long word for DNA is deoxyribonucleic acid. That's a mouthful, so it's totally fine to just call that genetic material DNA. Number 4 is that living organisms have adaptations. An adaptation is any modification that makes an organism better able to function in its environment. Evolution is a change in a population of organisms over time to become more suited to that environment. Or really, what's happening is that those living things are acquiring those adaptations and passing that genetic material to the next generation. And over time, we will see more of those adaptations within the population. So let's check what we know. The ultimate source of energy for life on Earth is, and you should choose D, the sun. Now may be a good time to pause the video and go back to your guided note page or the first part of this presentation and rescore how you feel about those objectives. And then take a minute to score your new objectives, explaining natural selection and evolution, and then distinguishing between domain bacteria, archaea, and eukarya. So let's start with evolution as a theory. It is going to explain the diversity and the unity of life. And the most famous player when we talk about evolution is Charles Darwin. And Charles Darwin came up with the idea of natural selection as the mechanism that causes evolution to happen. And the theory of evolution suggests that all living organisms descend from a common ancestor, or that there's descent with modification over time. Darwin's big idea, of course, is natural selection. And really, what this says is that there are some parts of the environment that are going to select which traits are better for that environment, and then those traits are more apt to be passed on. Really, what happens is that there's more individuals in a population than the ecosystem can support. Individuals with the favorable traits produce more offspring, and those offspring are able to survive, and then they're able to reproduce. And over time, this will increase the favorable traits in the population. Mutations to that DNA is really what fuels natural selection because it fuels variation in those traits among the members of a population, and then nature takes over and chooses thumbs up or thumbs down to the traits that that individual has, and over time our population will change. Here's a great example from your textbook. Here's a deer, and they eat these leaves. And as we see, there is a bunch of leaves that don't have spiky thorns, but then there's a mutation that gives this plant spiky leaves. And as we see, the deer likes to eat the smooth leaves, not the spiky leaves. So the deer is going to get rid of some of those smooth leaf plants, eat them up. They don't reproduce, but the spiky leaf plant does. And then over time, what we see are more leaves with the spiky spines because that is the best trait for that environment. We can model some of this evolutionary timeline by making a tree of life. And this evolutionary tree is really a family tree. It traces back the ancestry to a common ancestor. And then we see split offs that are because of these emergent properties, these new things that happened as an adaptation and then changed what our end result was. So we see there's a common ancestor and there's a split to bacteria and archaea. From archaea, it's a split to eukarya. And then that's a split to protists, which are different than plants, which are different than fungus, which are different than animals. What we see is that there are scientific names that help us to identify individuals within that tree of life. And the scientific names are universal, they're Latin root or Latin based, and we call it a binomial nomenclature because it's a two-part name. The first word is the genus, and we capitalize it. And the second word is the species, or it's also called the specific epithet, which is written in lowercase. And then both words are italicized. So a great example for humans is Homo sapiens, and we see the H is capital, the S is lowercase, and both words are italicized. If I was writing this in print, like I put it in my notes, instead of italicizing it, I could underline it. Same thing for corn, it's Zea mays. There's a really fun video linked in the D2L. It's this short SciShow where it talks about how scientists are able to name the creatures that they find. And so if you want to take a look at that, now would be a fun time to do that. And then feel free to think of what you would name a new living thing. We then want to organize that binomial nomenclatured set of things. So we have taxonomy, and that's the discipline in biology that identifies, names, and classifies living organisms according to some rules. There's also systematics, which is studying the evolutionary relationship that is between those different levels of taxonomy. We are going to go from the least inclusive at the species level to the biggest level, domain, and that would be species, genus, family, order, class, phylum, kingdom, supergroup, and domain. I actually like to remember them in the opposite order, from the most inclusive to the least inclusive, or domain, supergroup, kingdom, phylum, class, order, family, genus, species. Supergroup is new, so I kind of keep it out of the naming, and I like to use the mnemonic device. Did King Philip come over for grape soda? And each letter of Did King Philip come over for grape soda reminds me to look at D for domain, K for kingdom, P for phylum, and so on and so on. If we take a look at these categories, what we see is that each category is going to include, if we go in this order, more than the one before it. So there's more living things in domain than compared to species. Another way to take a look at this is if we were to look at humans and corn. They're both in the same domain, eukarya, but then they immediately become something different, where humans are in supergroup Opisthoconata, corn is in Archaeplastids. If we then went down to kingdom, it's now different, right? Kingdom Animalia for humans, kingdom Plantae for corn. But that's still a lot of things that are in that group, right? If you think about Animalia, that includes humans, but it would also include lions and dogs and octopus. Same thing for plants. It's including corn, but it's also including roses, and it's also including moss. So still many things are in those groups. As we get closer to species, it becomes less of a large category. It becomes more selective, right? When we get down here to something like order primates, right? That's a much smaller group than just Animalia or just mammals. And then finally, we end at our one specific thing. For humans, it's Homo sapiens. For corn, it's Zea mays. Let's take a look at all the different domains. There's domain Archaea, which is going to contain single-celled, prokaryotic living things that like to be in very extreme environments. So what does all that mean? Unicellular is one single-celled. Prokaryotes are cells that do not have a membrane around the nucleus or membrane organelles, pieces that build up the cell. When we say extreme environments, we mean like super duper salty, and they're halophiles. They might live where there's a lot of methane, like in the guts of cows, so they're called methanogens. They can live where it's really hot, so they're called thermophiles. So they're probably similar to what we saw on a very early Earth that was hot and didn't have an atmosphere to protect it. And here is our great example of a type of archaea. They can use different chemicals to make their food, and they're kind of weirdos of our domains. They used to be grouped with other prokaryotes, and then in the 1980s, we realized they needed to be their own separate category. Then we have domain bacteria. That's going to be some more single-cell prokaryotes, but they live everywhere. You're probably familiar with this if you've ever had to take an antibiotic to get rid of a bacterial infection. They can do photosynthesis. They can use chemicals to make their food. Here's an example of E. coli bacteria, which are found in the guts of warm-blooded animals. Then there's domain eukarya, which includes single-celled and multi-celled eukaryotic things. Eukaryotes have a membrane-bound nucleus and organelles. Within domain eukarya, we see four kingdoms. We have kingdom Protists, which includes algae, and protozoans, or animal-like protists. This is kind of the junk drawer category. If you're not quite a plant, fungus, or animal, but you're single-celled, you kind of get dumped into this category. Then there's Kingdom Plantae, which is going to be moss and ferns and cone-bearing plants and flowering plants. Most of them are multi-celled, and they have cells that are able to do photosynthesis or make their own food from sunlight. Then there's kingdom fungi, which includes molds, mushroom, yeast, and ringworms. Again, mostly multicellular, except for yeast, which is single-celled, and they absorb their food. And then there's kingdom Animalia, which is sponges, worms, insects, fish, frogs, turtles, birds, and mammals. They are going to ingest their food. Again, if we're taking a look at our kingdoms, it is domain archaea. Kingdom, we would just say archaea at this point. We still are trying to determine different kingdoms. Domain bacteria, again, you probably would also just say kingdom bacteria because we are still trying to determine through DNA research where everybody goes. And then in eukaryote, we've got protists, fungus, plant, and animals, but we do have supergroups that is inserted between the domain and kingdom, and really it's because of protists and how strange they are that we realized we needed that supergroup designation. So this is not a slide I expect you to memorize in any way, but if you're really interested, there are some different supergroups here: Excavata, Chromiolata, Rhizarian, Archaeplastidia, Amoebozoum, and Episcoconata, and some examples. If this makes you really excited, then Bio 102 may be for you. We spent an entire chapter on learning different protists. All right, so back to some test your knowledge. Which of the following lack a nucleus? And that would be E, archaea. All right, now might be a great time to go back and double check your rankings for section 1.2 and then give yourself a ranking for section So let's talk on the process of science. We use the scientific method. And the scientific method is this very standardized set of steps in order to gain knowledge by research. And the scientific method can be divided into 5 steps: observation, hypothesis, make your predictions and do your experiment, collect the data and analyze it with statistics, and then the results and conclusion. So a good thing you can try right now is to kind of pause this video and see if you can put the scientific method into some sort of flowchart or order. And let's see what you get. Did you make something that looks like this? Probably not. Maybe you made one straight line, kind of like a food chain, but real science in the real world is a lot more complex and creative, where you'll have some sort of observation, some ideas on what you think are causing that thing to happen. You predict it, what will happen if you test out one of these things, design that experiment, and then maybe you find out that wasn't it. But it's not like you say, well, we did science for the day and we give up. No, you have more questions. And so then you'll have your next hypothesis to test, and you'll come up with your more predictions and experiment some more. And you might find yet again that you reject that hypothesis. And so we've got to try something else. And we come up with a few different experiments for that something else. And we finally get some sort of, predictions being confirmed. Like, oh, we thought this would happen and it did. Now you can come up with your conclusion. But again, it's not like it ends at conclusion and you say, yay, I won science. You usually have a lot more questions that you want answered. So this is more like what our scientific method looks like than just a straight line flowchart. So, of course, your observation is when a scientist uses their senses, which again is something like sight or smell or hearing or taste or touch, to just observe something that is happening in the world. And then they'll come up with a hypothesis, which is really an educated guess or a tentative explanation as to what is happening. And a good example that is in your textbook is from discovering penicillin. It was this fungus that was growing on oranges. And at the same time, while that fungus was growing on oranges, they weren't being decomposed by bacteria. And that was noticed. And from that, we develop penicillin. And we say that these hypotheses are developed through something called inductive reasoning, or that you have some knowledge from being a person in the world. And so you're relying on those senses and your knowledge to create this testable hypothesis. Then, of course, you'll design an experiment with procedures that is designed to test that hypothesis. And of course, now we're using something called deductive reasoning because we want to get to an end result. So we're making a prediction and designing an experiment hoping to get to that result. And then the matter in which a scientist does this experiment is called the experimental design. And a good experimental design ensures that we're only looking at one variable called the independent variable or experimental variable. And we're only testing one experimental or independent variable at a time. Either vocab word is totally fine to use. I like independent variable because it's the thing that I change. I manipulate this variable in my experiment. And I like that letter mnemonic. I for independent, I for I change it. There's often a test group that we expose to that change or experimental variable, and a control group that goes through the experimental process, but they're not exposed to that experimental change. If the control group and the test group have the same results, well, then our educated guess wasn't correct, and we need to go back and try something else. The data that we collect is the results of our experiment. It should be observable and, if we can, a numerical thing that we're measuring. And it should be objective or unbiased as possible. We can then use tables and graphs to illustrate our data. We also use statistics in science. So we're going to analyze that data and we're going to measure variation. There's something called standard error, which is how far off of the average is each point in the data. And we can use that to measure something called statistical significance. It's also called the p-value or probability value. And for most experiments, when we're comparing our control and our experiment, we want a p-value of less than 5% or 0.05 because that shows us that 95% of the time, the results are because of what we did, what we changed. And only 5% of the time is it because of probability, that's how it would just happen to turn out. Medical testing, their p-value is usually 0.01 or 1% is the acceptable probability chance. And it just depends on what you're trying to test that you can change that p-value in order to, satisfy that result. But generally accepted is a 5% due to chance. And then we could graph something like this, where we would show our x-axis on the bottom here, that would have our independent variable, and then our y is going to be our responding variable, or what we measured. And then these little bars are our standard error. And then we can plot exactly what data we collected, or in this case, it's the average blood cholesterol each week with these little dots. Then, of course, we do conclusions. That is when the data is interpreted and determined if our hypothesis was supported or rejected. If the prediction happens and it was correct, we say we support the hypothesis. If it is not, we reject the hypothesis, and then we would come up with new ideas for study. And then the main goal is to report what we found to scientific journals. And then science peers actually review those articles before they're published, and other scientists may even attempt to duplicate or dismiss what you did in your experiment, all for the betterment of science. And then, of course, scientific publications is once we have that peer review, we're publishing this data. Not all studies are accepted. They have to be peer reviewed to be accepted. But of course, this is not the source where most people are finding their scientific knowledge. They're finding it from secondary sources. So there will be a science communicator whose job is to read these scientific publishings. And truly, they're not the most exciting things to read. They're very technical. And then they'll create a piece that goes in, a documentary or a news article that's a little more digestible by the general public. So if you are in my in-person class, we do these minute papers as our participation grade. If you're in my online classes, this is a nice place to just think about it. So you're going to have a minute-ish to jot down all of your ideas on this question. Then you'll talk to your peers, and then you're going to turn in what you wrote. For my in-person classes, this is under the assignments tab. For my online classes, you don't have to turn this in. So what is the advantage of peer review in science? This might be a good time to pause the video. And then to come back to it, an advantage of peer review in science is that you have other scientists verifying that what you have claimed really is happening. And that is eliminating any sort of bias as much as we possibly can. Because many eyes are on these scientific findings before they're put out to the public. And then, of course, a scientific theory is a concept that's going to join a bunch of related hypotheses together. There's a lot of data behind it. And it means that, it's been around a long time and has a lot of tests. That doesn't mean it can never change. Science has backed some incorrect theories in the past. And then we have to change when the data changes and our conclusions change. Something that is a much more widely accepted thing is called a scientific law or principle. It encompasses a lot of different theories, and really there's no challenges. There's no data that we're probably going to find even with changes to technology. It's something like gravity, right? Every single time you drop something, it falls to the ground. It never floats away. That would be considered a law. So some theories that are in biology, we have the cell theory, we have homeostasis, we have evolution. All of these have many different experiments from many different fields of science, and that data is all supporting these theories. And again, science has backed some incorrect theories in the past, and then when new information is presented, we have to update what we believe based on where the data takes us. So let's take a look at a study together. We have a group of people and they all have stomach ulcers. And we have 2 antibiotics that can be used to treat these ulcers. And the one that's on the market that is used is antibiotic A, and antibiotic B is brand new. And our hypothesis is antibiotic B is better for treatment than antibiotic A. So we're going to take these people that have stomach ulcers and we're going to split them into 3 groups. One group is my control group. They receive a placebo. That's a pill that doesn't have any antibiotics in it. And they're there to compare our results. And we want to treat them like they're in the experiment, but we're not going to tell them they're not getting the antibiotic. Then we've got test group 1, which is getting antibiotic A, and test group B that's getting antibiotic B. We're going to give them this medication, and then we're going to do an endoscope to see their stomach ulcers. And then we can graph our data, and we see that control group, only 10% of the people were cured of their ulcer, 60% in group A that got antibiotic A, and 80% in group B that got antibiotic B. And we would say then that antibiotic B is better at treating ulcers than antibiotic A. So here is that exact study in words. If you want to read through these. And now a little sort of check your understanding. This comes out of your textbook. So you may want to pause the video here and try to match the letter A, B, C, D, E, F to where you think it should go on this flow chart, reading this little blurb. And then, of course, your book provides you an answer key. And then we're at our last section, so you may want to go back and rank yourself on section 1.3, and then of course give yourself a ranking for section 1.4 about science and technology and some challenges facing science and society. So if we take a look, science is this systematic way of getting knowledge about the world, right? We use the scientific method to do so. And technology is that application of that scientific knowledge. And it's going to focus on what humans are interested in doing with that knowledge. So some examples of some technologies might be cell phones or new medications. And then, sort of a quick check here. You may want to pause. What are the challenges facing society and science? What technologies do you know that might help? So, one major thing that is facing science today is climate change. That refers to the change of the normal cycles of Earth's climates because of human activities. And it's caused by an imbalance of carbon cycling. What we see is there is natural fluctuation to that level of carbon, but since the Industrial Revolution, that level has gone higher than we have on any recorded record, even dating back thousands of years in ice core records. And we think that this is because of the burning of fossil fuels, along with destroying forests to replace it with farmland so that we can, feed our growing human population. That increase of CO2 is going to cause heat to be trapped on our planet in our atmosphere, and that is called global warming. And it is this greenhouse effect that allows us to have life on Earth because we have liquid water, but too much heat is causing temperatures to change. And places that were once really cold are now warmer. Perhaps if you were warm before, now you're like a desert. It's making storms stronger. It's melting ice. We have drought. And this is all something that, we are facing as a society. Biodiversity and habitat loss is another challenge. Biodiversity is the total number of species and their abundance. It also takes into account the variability of their genetic code and the ecosystems that they live in. And we think that there could be 8.7 million species, but we've really only identified and named about 2.3 million of them. And when we deforest, when we overfish, when we destroy habitats because of climate change, it can lead to extinction, which is the death of the last member of a species or a larger classification category. And we're losing hundreds of species every year. Some scientists estimate that, we are losing species at a rate that is equivalent to the last mass extinction, which is when dinosaurs went extinct. Destruction also has some unintended effects for people. We depend on ecosystems for food and medicine and materials. When we drain wetlands, like in Mississippi, in Ohio, in Charleston, it actually causes worse flooding and ruins farmland. When we destroy rainforests, it decreases our lumber supply and it kills species, some of which we use to make medications. And then, of course, there are emerging diseases. So, over the past several decades, we've gotten new diseases like different kinds of flu, like H1N1, or SARS, or MERS, or even COVID. And where do these come from? Well, as the climate changes and as we deforest and biodiversity changes, we're now coming into contact with animals and plants we never had before. And when we come into contact with those plants and animals, it has the possibility to spread disease. We also have changes in behavior for both the plants, animals, and people. The use of technology can spread disease. Legionnaire's disease was actually spread through air conditioning vents. Before we had air conditioning vents, we didn't have Legionnaire's disease. Globalization, or being able to have this very globalized society where you can hop on a plane today and by tomorrow be in Europe and then hop back on a plane and, go to Asia or Australia and hop back on a plane and come back to the United States. That is putting people in contact with each other, and that spreads disease. And then, of course, those disease causing things called pathogens, they mutate and they change hosts. So, something like avian flu was affecting birds, but now it's jumped to people. So, whew, good work. You've made it. Now is a great time to check out your chapter study guide, check out your objectives in this chapter, check out that extra resource folder. Make sure that you've completed your McGraw-Hill Smart Book and that you're studying this material for your quiz. And then, of course, anything you want to do to prepare for the next class. Thank you for watching our video, and as always, if you have any questions, please reach out.